Let's wrap up this genetics chapter with a discussion on genetic modification and biotechnology. Some really important and recent advances in gene technology that you should be aware of include the PCR reaction, DNA profiling, gene sequencing, gene transfer, and cloning. All right, so the ultimate goal of doing the PCR or polymerase chain reaction is to get a situation where we have enough DNA to profile it. But DNA is really small, it's super skinny. So in order to visually see it, we need a lot of copies of DNA. So oftentimes you'll hear about PCR as something that amplifies DNA. It doesn't mean make it bigger, it just means make a lot more of it. And if PCR is sounding familiar to you, good job. Okay, polymerase is that enzyme that's involved in DNA replication that helps to build complementary strands onto parent strands of DNA. Now, to get a product that looks like this, you have to go through a process called this. Okay, so gel electrophoresis is a process that's going to separate DNA into bands, okay, or DNA segments of different lengths so that we can create this, which is called a DNA profile. So actually the first thing that we're going to do is we're going to use PCR to make a crap load of copies of DNA. But then once we have all those copies, we're going to use enzymes to cut the DNA into different sized fragments. So if I have a long strand of DNA, okay, I may cut it into different sized fragments. Okay, and then those different sized fragments are all going to still be in one solution. And I'm going to take that solution and I'm going to put it into a well at the top end of what we call a porous gel. Porous meaning has a bunch of holes in it. So inside this gel are a bunch of minuscule microscopic holes. The end with the DNA sample in it is going to be exposed to a negative electrical charge. Now, DNA is also negatively charged, so when you expose the negative DNA to a negative electrical charge, this DNA will want to get away from that very fast, okay, and it's going to migrate through the gel. But because we have things of different sizes, they're going to make it different distances through the gel because of these holes, right? So things that are really small are going to go farther down, like this guy. Things that are really big, like this guy, are going to get stuck up here at the top. And so that's how we end up with different bands here. So each one of these bands represents a segment of DNA, so kind of like this, okay? So a segment of DNA that was cut from the original strand, but made it different distances down through the gel. So what determines how that DNA moves through the gel? Well, the first thing is charge, right? So DNA has a negative charge, which is why we have to place a negative electrical charge or negative electrode up at that end to push that DNA. And the second thing, of course, is size. So the smaller fragments are going to move farther down, and the larger fragments are going to get stuck here up at the top. And so when we get that banding pattern in DNA, we're going to see an image like this. And this is what we call a DNA profile, also known as a DNA fingerprint. And that's because they're unique to each individual. So if I have a person whose DNA gets cut up here, here, and here, another person's DNA might get cut up into different length segments. Okay, and so therefore they're going to have different banding patterns because they'll have different length of DNA moving through the gel. So a perfect match in the banding patterns means that someone has identical DNA. A partial match like this one would most likely belong to someone related. Okay, so for example, a child is going to have perhaps, and let's see, this might be the child here, DNA from the mother, okay, as well as DNA from the father. So establishing paternity is definitely one of the uses of DNA fingerprinting. So again, a baby is going to inherit some things from the mother and inherit other things from the biological father. All right, so another thing that we can use these DNA fingerprints are comparing DNA at a crime scene with potential suspects. So if you're asked this question on the IB exam, you have to explicitly say these two terms. You can't just say used at crime scenes or used to identify people. You have to say matching DNA left at a crime scene with potential suspects. On the other hand, a lot of work has been done recently to release prisoners that are wrongly convicted. So we still have many people in prison that were convicted before DNA science really became readily available. And so now that we have the ability to test their DNA, we could potentially release people who don't belong there. And the next cool thing that's really going on right now is to establish relationships, okay? So it's really cool to analyze the DNA to take a look at migrating patterns or evolutionary relationships between individuals. So we would think that two individuals that are close to each other geographically, like maybe here and here, might have much more similar DNA than two individuals that are here and here. Also, two organisms that have a more recent common ancestor or a closer evolutionary relationship would have more DNA in common than two organisms that aren't. All right, so let's interpret one of these things. Let's say we have a crime scene and a known victim, and we also have two suspects. What we need to do is to try to match DNA at the crime scene to one of the suspects. Now, we know that this DNA that was left at the crime scene isn't the victim's. Look, none of the banding patterns match up. So we can eliminate that person, okay, from being the donor of this DNA. I think we can also eliminate suspect number 2, right? So the banding patterns just don't match up in every single place. But suspect number 1 over here definitely looks like she might be in trouble because all of these banding patterns, okay, have equivalent patterns over here at our crime scene. So suspect number 1, okay, would be our match there. All right, so let's move on to another concept using genetic technology, which is gene transfer. Now, I love this term gene transfer because it's exactly what it says it is. It's taking the gene out of one organism and transferring it into something else. And this is really cool that we can do this, and it's all because of the universal genetic code. So when we say the genetic code, we mean that the codons in one organism make the same amino acids as in every other organism. So the codon ACG makes threonine in butterflies. It also makes threonine in people, also makes threonine in bacteria. So it doesn't matter. And that's why I can insert genes from one organism into another, and that organism is A-okay with it because that DNA works the same way and makes the same amino acids as the rest of the DNA in its genome. Okay, now let's say I want to take a gene out of an animal cell and put it into a bacterial cell just for fun. How do I do that? All right, well, the first thing I need to do is get the DNA out, right? So I want to get the DNA out of the donor cell and also remove the DNA from the cell that I want to insert it into. All right, then in step 2, I literally want to take like little genetic scissors and cut out the gene that I want to insert into our donor cell. Okay, I also need to cut a space in the host cell's genome so that I have room to put that in. And to do that, we're going to use an enzyme called an endonuclease. Okay, this ASC here is the key word for remembering that that's an enzyme. We also call them restriction enzymes. You may see them written either way. So endonucleases, restriction enzymes, I'm going to cut out that segment of DNA. All right, the next thing I need to do is take this gene I cut out and stick it in here. Okay, to do that, I'm going to need an enzyme called ligase. Okay, so my higher level friends, you should remember this one from our discussion on Okazaki fragments and DNA replication. Okay, so ligase is an enzyme that will literally take this gene and glue it into this chromosome. So right here, it's going to stick it in there. All right, and the last thing I've got to do is I've got to take this modified chromosome and stick it back into the host cell. So now at the end, if I've done this correctly, I have a human gene now sitting comfy and cozy inside of a bacterial cell. Now, I say human and bacterial cell because there's an absolutely awesome and cool application for that. And that has to do with the gene for insulin production. So we know that some individuals, specifically those with type 1 diabetes, whose beta cells in their pancreas just don't produce insulin, are in needs of large doses of insulin. And before, we would have to get those from animals, like harvest them from pigs. Now we can get insulin by making bacteria produce them. So I will tell you now, we use the E. coli bacterium, and E. coli don't produce insulin on their own. Okay, so what we do is we cut a hole in the E. coli's genome, and into that big circular chromosome, we insert the human gene for insulin. Then when we put that back in and allow that E. coli bacteria to reproduce, it produces a whole colony of bacteria that now have that insulin producing gene. The E. coli bacteria don't know they're not supposed to produce insulin. They think, oh, it's in my chromosome, I better do what it says. And they end up producing a bunch of insulin, which we can then purify and give to people who really need it. All right, so let's talk about some terms we can use for organisms whose DNA we've altered. All right, one that you've probably heard of before are GMOs, genetically modified organisms, and those are specifically an organism with a gene that has been artificially inserted into its genome. And we've done that to give it some kind of a desirable property that it didn't have before. We also can call these organisms transgenic. So trans meaning across, so across species, and then genes, of course. So any organism that has a gene from a different species into its genome. So you may be asking yourself, what the heck is the difference between those two words? Well, when we say genetically modified, that could also include removing a gene. It could also include altering a gene. So it doesn't, and I probably shouldn't have included this here, it doesn't have to necessarily be limited to artificially inserting a gene. If we're just modifying it, we could also be just changing or removing a gene. Transgenic, on the other hand, actually has a different gene from a different organism inserted into its genome to give it some kind of property it didn't have before. All right, so let's talk about a couple applications of this transgenic movement here. So we're going to start with corn. And original corn was very susceptible to insect larvae, and corn farmers would often lose a good amount of their yield to damage caused by these insects. So we can take a gene from a bacterium and insert it into the corn's genome, and that's where we get this term Bt corn. The B and the T come from the genus and species name of that bacterium, and that bacterium had genes to kill insect larvae. So corn obviously couldn't do that on its own beforehand, but now we've inserted that gene in there, and so no insect larvae are able to get into the corn, eat the corn, and destroy the corn yield for the farmer and the consumer. Another really cool example that's pretty common are with tomatoes. And tomatoes are very fickle plants, okay? They're kind of like my grandma. They're not really comfortable outside of their temperature range that's pretty narrow. Specifically when it comes to freezing. So when we get cold temperatures, if your tomatoes are already growing, you're in big trouble. They're just not going to last. Well, along comes this ability to take genes out of this Arctic flounder and put it into a tomato. And this gene happens to produce almost like an antifreeze type of situation where it makes these tomatoes more cold tolerant. So now they can be grown not just near the equator, but farther and farther away from the equator, making fresh local produce a little bit more readily available during longer growing seasons in the year. All right, so good for farmers, maybe good for consumers. Also, an interesting, like, humanitarian effort on the part of geneticists. So, we can do this really cool thing to rice by inserting a gene into its genome to produce beta carotene. And beta carotene is what makes carrots orange, okay? And so, just like it makes carrots orange, it makes the rice this goldeny color. We call it golden rice. And beta carotene is something that your body has to have in order to produce vitamin A. So countries that were really reliant on rice, like for a staple crop, were largely deficient in vitamin A. Like they were just, or beta carotene and therefore vitamin A, and they were having a lot of problems with blindness. And so the percentage of children, especially who were experiencing blindness and vitamin A deficiencies, was just incredibly high for those countries that were depending on this regular rice. So with very little economic input, with very little monetary funding, we can simply alter the genome of that rice to produce golden rice so that this kind of deficiency doesn't have to happen anymore. So in our last discussion, we talked about hemophilia, okay? And hemophilia affects people, a lot more males than females, who inherit a recessive copy of a gene that doesn't allow them to produce one of their clotting factors called factor 9. And so without that clotting factor, oh boy, it's really hard to stop bleeding. So one of the things that geneticists have figured out a way to do is to produce that clotting factor in sheep, okay? And so this is a great example of a transgenic, not plant, but animal. So the principle is the same, inserting a gene into a genome that wasn't normally there. Now you have to do this at the embryonic stage in order for all the cells of the organism to have that gene. So what scientists did is they found a way to take the human gene for producing this clotting factor and stick it into the genome of a sheep embryo. And then when the sheep makes its milk, okay, it has that clotting factor in the milk, and then people with hemophilia can then drink that milk. So why didn't they just do this to humans? Well, it's a little bit of a shady ethical situation to be messing around with the genomes of embryos and then using those embryos to specifically produce things. So for right now, we're having a lot of success using it in these transgenic animals like sheep. Now, I think it's naturally for our minds to wonder from all this genetic technology to the ultimate kind of genetic miracle, which is cloning. But the way that we think about cloning isn't necessarily as like cool and mysterious as you might think. So a clone technically is any offspring that's an exact copy of its parent. So you have to remember that any organism that reproduces asexually are always clones. So there are a few plants that do this, a lot of unicellular organisms like bacteria. Okay, they're always clones. However, there are some plants, okay, that can either reproduce sexually through their flowers or have the ability to clone themselves. So they can do either or, okay? And they do that cloning process through something like this, like a shoot. And a great example of a plant that can do this is a potato plant. So potato plants, like this one, can, like I said, either reproduce sexually or asexually and just make a clone. So to do that, if you want to make a clone of a potato plant, you can do that. You just take a little stem, a little offshoot, and you can plant that. And you will get a brand new but identical plant. So this is an advantage, right? You don't have to worry about fertilizing these flowers or relying on pollinators. If you have a really cool, awesome, like rockin' award-winning potato plant, you can make some clones of that plant. So a lot of advantages to being able to do that. But of course, there's some disadvantages there too. And in particular with potatoes, we saw this come to light and actually decimate almost an entire population. So when we clone something, That means that all of our organisms are exactly the same. Okay, so if I produce a whole field of potatoes that were all cloned from one plant, all of my offspring are the same. Now, that's okay unless something in the environment changes, like, for example, blight. So, blight was a fungus that hit the potato fields of Ireland and killed all of the potatoes. Okay, a large amount of the potatoes that were planted there were planted by cloning, and so they were all genetically identical and therefore equally susceptible. So it left people literally without food to eat. We're talking about 1 million people dying of starvation. 1 million is a lot for a relatively small country to begin with. So when we clone things, what we're really missing is this genetic diversity. If you have a diverse population, if something in the environment changes, then perhaps it kills some individuals, but because you're genetically diverse, you might have some offspring that can survive. So some definite disadvantages to cloning that need to be considered, especially in terms of agricultural use. So cloning in the animal world is actually incredibly rare. We know that like vertebrates can do this, right? But there is one invertebrate animal that can clone itself, and it's called the hydra. And hydras look like this. They can either sexually reproduce or in times of need, they can do a process called budding, which a little mini-me pops out, and it again is a clone of the original parent. It's not necessarily an advantageous adaptation for things like larger animals, vertebrates, etc. Again, because you're really missing out on some of that genetic diversity when you go through cloning instead of sexual reproduction. So we know cloning happens in plants. We know cloning happens in some animals. What about in humans? Well, yeah, but not on purpose, okay? So clones were really kind of first recognized when we were looking at identical twins. So how do we make identical twins? Well, we start with a zygote. So here we have the sperm nucleus, so the sperm DNA, and the egg DNA, and they end up fusing together to make a zygote. Well, that one zygote will copy, and so all of these cells are identical. And then for some reason, okay, this ball of cells splits into two, okay? And so one ball of cells will become its own embryo, and the other one will become its own embryo, but because they both originated from this single zygote, they are identical. So again, it's important to note that these are undifferentiated, okay, and they have identical DNA, and for some reason, they split, okay, and they end up making identical people. All right, now let's say you want to make a clone of something, but they're already old and their cells are differentiated and you can't turn them back into an embryo to clone them. How do we do that? Well, that's something called reproductive cloning, and we have another name for it called somatic cells transfer. I'll tell you why in just a moment. But that's when we take an already differentiated cell and we use it to make a new individual who is a clone of that original parent. And this was first done successfully in 1996 with this celebrity named Dolly the sheep. So how do we do that? Well, a somatic cell, so this is just a differentiated body cell, like a skin cell, liver cell, etc., was collected from the sheep that we wanted to clone, okay? And we removed the nucleus, so we took out all the good stuff. So we took out all the DNA. We then found another sheep, and we collected an egg from her. We then removed the nucleus from that egg, but left all the other things like the organelles and all the other stuff needed to turn that egg into a zygote. And we fused them together. So we took the nucleus from the sheep that we wanted to clone and the empty egg from another sheep, and we fused them together to make a different egg. So now let's think about what we have. Okay, so we have an egg that's ready to go with a diploid nucleus. You see, because we took that from a somatic cell, it's already diploid. It is already got all the chromosomes that it needs. It doesn't need a man, doesn't need a sperm. It already has all those chromosomes. And we let that egg multiply and go through all of the embryonic development that happens in those early days. We then took that embryo and stuck it into a donor sheep, just like you would do during something like IVF. The sheep has that baby, and the baby is an exact copy of the original sheep because it has that DNA. So cool. Now, Dolly the sheep had a lot of problems, okay, a lot of medical problems, and there are some interesting ethical debates to be had with reproductive cloning. And sometimes scientists just aren't interested in making a whole individual, they're just interested in making one type of cell. And that's what we call therapeutic cloning. So therapeutic cloning, we're still taking undifferentiated cells from an embryo. Okay, so again, still some ethical gray areas here, but we can expose these cells to different hormones and cause their development or differentiation to go in a certain desired path. So if I need to make new epithelial cells or intestinal cells or heart cells or bone cells, whatever kind of cells I want, okay, I can just take those embryonic stem cells, expose them to hormones, and direct them into new types of tissue. Why would I ever want to do that? Well, there are a lot of therapeutic applications here, okay? So let's say I've suffered a serious burn and I need to replace skin, okay? It would be great if I could take my own somatic cell, like my own DNA of something that's undamaged, take that nucleus out, put it into an empty egg from a donor, okay, and then cause that to develop into an embryo, take those embryonic stem cells out, expose them to hormones that are going to make them into skin cells, and now I have skin cells that have my own DNA, they're just like the rest of my skin, that I can use to replace a burn, or a heart, or a kidney, or any other kind of tissue. And again, these are really interesting because they would have my own DNA. So some obvious benefits here, replacing damaged tissue, reducing the need for organ transplants. So if I have kidney failure, wouldn't it be great to just be able to grow new kidneys that have my own DNA using this method instead of waiting and potentially dying while I'm waiting for an organ transplant? And reduced medicine use. Okay, so if my organs are failing, even if I get a new organ, then I'm going to have to be on immune suppressing drugs for the rest of my life. So it could cut down on a lot of medicinal stuff. On the other hand, there are some serious ethical concerns to be considered. So first of all, this embryo that we're creating is not ever going to develop into a child. When you remove these cells, that then becomes non-viable. And there are some questions to consider over whether destroying a potential human life is an ethical move or not. All right, so speaking of controversies, let's go back to talking about GMOs for a moment. So GMOs are scientifically ridiculously cool, okay? From an application standpoint, there's also some really good things going on. Like I can insert genes into plants that cause them not to need pesticides grown on them. So the crop yield goes up from less insect damage. Like the golden rice, I can add nutrition. But there are also a lot of really important questions that we need to ask ourselves in terms of some long-term consequences. So for example, there's been some research to suggest that native species are being affected by these genetically modified species right next to them. I'll talk about an example in a moment. There may be some ethical concerns there as to whether or not as humans we have the right to alter the genome of other organisms. There are some huge patenting concerns, like who owns those new plants? Am I allowed to make them breed? Okay, is Monsanto going to sue me and take everything that I own? Perhaps. Okay, so a lot of legal things that we haven't caught up on. Okay, there are many issues with potentially killing pollinating insects. So like the pollen from the GMOs is detrimental to pollinating insects like bees. It's one of the things that we think may be affecting our current bee population. And really what I think is most interesting from a scientific point of view is that we have no long-term data. We don't know what the effect of eating GMO crops for 50 years is because they haven't been around that long. So we go back to this precautionary principle, which says that we have the obligation to prove that something is safe, okay, before we put it out there on the market. And really, we just haven't been able to do that with GMOs because we don't have enough long-term data. Okay, so let's go back here to the beginning for a moment. Okay, I want you to do this one on your own. Okay, so you can interpret this DNA fingerprint to determine the paternity. So here's the mother, here's the offspring. I want you to figure out which of these males is the father using the banding patterns of this DNA profile. Okay, now I told you we'd cycle back around to talking about the use of GMOs like the BT corn on native species. So I want you to take a look at this graph, and you can do this interpretation on your own. This is just a graph. This isn't so hard, okay, to take a look at how larvae placed near normal fields in BT cornfields differs. All right, so let's talk about why that difference may be taking place. So here I have a field of BT corn, and right next to it, on the edge of these cornfields, I often find a plant called milkweed. It's exactly what it sounds like, okay? It's a weed, and it's not all that pretty, but it happens to be the primary food source for these beautiful monarch butterflies. And so when these butterflies are migrating, they're not only depending on this milkweed for a food source, but they are also laying their eggs on the leaves, and then their larvae, these caterpillars, often eat the leaves, and then of course they're eventually going to hatch into monarch butterflies also. So when we have this BT corn or any corn right next to the milkweed, oftentimes pollen from the corn will land on the milkweed leaf. We think that the BT corn pollen is somehow inhibiting the growth or survival of these monarch butterfly larvae. And so the regular pollen doesn't seem to hurt them, but the BT pollen causes these guys to die off at a pretty significantly higher rate. And so kind of one of the interesting impacts of genetically modified organisms that we don't often think of. All right, and we're going to end up here with our discussion talking about reproductive cloning. So reproductive cloning, remember, is like what Dolly the sheep was doing. And Dolly the sheep, the younger one, experienced a lot of medical issues that were not common in young sheep. So at a young age, this clone had problems that were similar to what older sheep normally produce. And so what we have to remember is that any mutation in the DNA that accumulated over the lifetime of the original donor sheep was then passed on to this young clone. And so perhaps the accumulation of all those mutations and that old DNA per se caused a lot of premature problems in Dolly the sheep. So in terms of health problems, there's certainly some things to consider. And then there are also some ethical things that we should really take a close look at as well. And that'll do it for chapter 3.5 on biotechnology.